Okay, so today's review session is going to be on DNA and genetics, which is what was covered with Ms. Tyson on Tuesday. So we're going to go through some questions that go over DNA and genetics. So the first question I have for us to start with is this question. It says, hemophilia is a sex-linked recessive trait. Which of the following describes the probability of hemophilia in the offspring of a man who does not have hemophilia and a woman who is heterozygous carrier? So what do we think the answer might be for this one? And this might take a little bit of work on the side. Okay, Rachel says C. Anyone else have an option? All right, well, it's actually going to be D. And the reason C is not correct is because it says 25% chance that their sons will have hemophilia. So we're just looking at the sons and not all of their possible offspring. If you were looking at 25% chance that their offspring, meaning male or female, will have hemophilia, then that would have been correct. But because they just want you to focus on the sons, it's actually going to be D, and that is 50% chance. So let's take a look at that and how that Punnett square works. So remember that dominant alleles or genes are going to be represented with a capital letter. So for this instance, we're going to put in a capital N. Usually it's the first letter of the actual gene you're looking at. But so that's why I chose N for normal, because normal is actually the dominant gene here. Recessive is represented with the lowercase letter. So in this case, the lowercase n is going to represent the hemophilia. And this is a sex-linked gene. That means it's going to be on the X or the Y chromosome. In this instance, it's on the X chromosome. So we need to remember that a male is XY and a female is XX. So we have the dad. It said that the dad was, let me go back here, the offspring of a man who does not have hemophilia. And I actually have this incorrect. That should be a capital N. I wrote that wrong when I made the slide. I apologize for that because he did not have it. And the mom was a carrier. So when we combine these together, actually the female here, the first one should be homozygous dominant, so she's normal. And this female is going to have a normal as well. Okay. When we go over here to the other offspring, if we bring the X over from that mom, this male will be normal. But if we bring the carrier X from the mom over, the male will have it. And so this was actually a smiley face. I made that mistake. I apologize. See, sometimes teachers make mistakes too, but I caught it, thankfully. Still doesn't change the answer, though, because, again, the answer is out of the males, which are only these right here, out of the males, it's a 50-50% chance that the males or the sons will have hemophilia. Okay? If we were looking at all of the offspring, it would have been a 25% chance, so that's why C might have seemed correct. But because they said 25% of females, then that's why it's not, okay, because it wasn't just looking at, or it said, sorry, 25% of the boys or the males, I believe. So it's not just 25% of the boys, it's a 50-50 of the boys. It's 25% of the overall all offspring. Does that make sense? Okay. All right, then let's move on to the next question. So this one's talking about genes, and so genes from medically important proteins can be cloned and inserted into bacteria as shown in the diagram below. So what we have here is a plasmid vector that the DNA fragment from a human gene is stuck into. So now we have, and the plasmid is basically DNA, okay? So now we have a human gene inserted into that plasmid DNA that we put inside the bacterial cell. so that the bacterial cell will now actually read that DNA and make copies of it and actually make some of that gene. So why can the bacteria recognize a human gene and then produce that human protein, even though it's a bacteria? Is it A, DNA replication in bacteria and humans is the same? B, bacteria cells contain the same organelles as humans? C, the basic components of DNA are the same in humans and bacteria? Or D, bacterial cells and human cells contain the same kind of chromosomes? Which one of those do you guys think it is, as to why we can do a human gene into a bacterial gene and it work? Got a C. Anybody have any other guesses? Another C? Okay, excellent. Perfect. It is actually C as the correct answer. So let's go through the other ones as to why they're not. So DNA replication in bacteria and humans is the same. That is not correct because in humans, replication is going to involve chromosomes that are going to have to be split apart and copied before division, things like that. But in bacteria, the cells actually do a binary fission, and it's just a single chromosome that has to be replicated, so the process is a little bit different. Bacteria cells, B is not correct because bacteria cells do not have the same organelles as human cells. Remember, bacterial cells do not have any membrane-bound organelles because they are prokaryotic, whereas human cells are eukaryotic. And D is not correct because bacteria cells and human cells contain the same kind of chromosomes. Again, that's not correct because bacteria cells just have one circular strand of DNA, and human cells are going to actually have the complex chromosomes that are wrapped around histone proteins to the DNA so that it can form that chromosome structure. So C is the correct answer. The basic components of DNA, those nucleotides with the nitrogenous bases, are the same in all species. Humans, bacteria, plants, they're all going to be the same. And that's because of the central dogma theory. And that is that DNA is the primary genetic source. And from that, we are able to transcribe to messenger RNA so that the proteins can be made through translation. So DNA is the main genetic source for all organisms, and all nucleotides are going to be built and look the same. All right. Next question is, the allele for brown eyes is dominant to the allele for blue eyes. Which of the following best explains how two brown-eyed parents could produce a blue-eyed child? A, each parent must be carrying the recessive allele for blue eyes. B, eye color is sex-linked trait and male children could only have the allele for blue eyes. C, mutations after fertilization could alter gene sequences and change alleles. Or D, one parent must have had only blue-eyed parents. So which of those would be the best explanation as to why two brown-eyed parents could produce a blue-eyed? A, excellent, perfect. That one didn't confuse any of you. Each parent must be carrying the recessive, because remember, a recessive can only be shown when it is combined to itself. Otherwise, it's hidden by the dominant. So if the both parents are hiding that recessive, there's the chance that the recessive can come together in the offspring. Now, keep in mind, I'm not very pleased that this is a question because brown eyes and blue eye color in general, especially for humans, is not controlled by one single gene. And that's what this makes it sound like there is an actual gene for brown eyes and a gene for blue eyes, and that's not true. Eye color is controlled by multiple genes that come together in certain ways to produce certain colors. So this is simplifying it down just to give you an idea of how dominant overpowers the recessive, and that's perfectly fine, but just keep in the mind in the back of your head that it's not as simple as just one gene for eye color. But here's, if it was, then here is the Punnett square that would back up that answer. One parent is on the top, so we have a big B representing the brown eyes and a lower B representing the blue eyes. So if a parent is hiding that recessive B on the male, and the female is also hiding that recessive B, we can see that we could have a 25% chance of a child getting blue eyes from that. Okay? And keep in mind when doing the Punnett squares, I have on both the Punnett squares I just recently showed you, I put the male geno on the top and the female genotype on the side. That can be switched if there's no exact placement. The male can be on the side and the female can be on the top. Does not matter. So just remember that that can be interchanged. And this type of Punnett square, the monohybrid cross, is what we're seeing here, where we're just looking at one gene at a time. Okay? All right, next question is talking about polydactyly. So when an organism has more than 10 fingers or toes, the condition is known as polydactylism. Although polydactylism is rare, it is a dominant trait. So that's kind of interesting, right? Just because something's rare doesn't mean it cannot be dominant. Dominant is referring only to how it is expressed, not in how prominent it is in the population. So remember that as well. So if two cats that are heterozygous for polydactylism mate and have a litter with a total of 12 kittens, how many of them would you expect to have more than 10 fingers or toes? So we have to do the Punnett square here so we can see what the percentage is. Ooh, that's a bad square there. So I got a couple of you already saying C. Let's just double check this just to make sure. So here's our Punnett square. If we want to use, I'm going to use N's again because P's are hard to tell the difference between the uppercase and the lowercase. So we know that the capital is the having the polydactylism. So we know that both parents are heterozygous, so that means they have to have the polydactylism but be hiding the normal. And if we do that Punnett square, we would get 3 of the offspring possibilities having polydactyly, and 1 of the possibilities having normal. So 3 out of 4 is 75%. 75% chance of having the polydactyly. So if they have 12 kittens, 75% of 12 is correct as 9. That means 9 of them would possibly have the polydactyly. Okay, and then I just did that again on this next slide to kind of show you how that works. And in this case, I used F for fingers, I guess. Oops. All right, so 75% with over 10 fingers or toes, 25% with normal, so 75% of 12 is 9. Any questions on these so far? Okay, good. Looks like we're doing pretty good. All right, let's move on. This says pea plants. The allele for white flowers is recessive to the allele for purple flowers. In the generation of pea plants, 89 plants produced purple flowers and 31 plants produced white flowers. Which of the following is the best conclusion you can make about the parent plants of this generation? A, both parent plants were heterozygous for the white and purple flowers. B, both parent plants were homozygous for purple. C, one parent plant had white flowers and the other was heterozygous for flower color. Or D, one parent plant was heterozygous for flower color and the other was homozygous for the purple. So what do we think would possibly give us an 89 to 31 split between the white and purple flowers? And we know that white's recessive to purple. All right, we got a couple different guesses here. We have one guess for A and one guess for D. So let's just kind of work through this. This is what's called a test cross, so you kind of have to work backwards. We know the offspring, but we don't know the parents. So first I would suggest figuring out what that ratio is, okay? So the easiest way to do that is to go ahead and just add up the total offspring, so 89 plus 31. And that's going to give us 120. And then you can find the percentage that those were. So 89 is what percentage of 120? So we can figure that out, and it's going to be approximately 75%. So 75% were purple at the 89. And then the 31, if you do find the percentage of what 31 is of 120, that's approximately 25%. It's off by a little bit, but it's pretty close. Okay, it's pretty close to that. So 25% were white. Okay, so we kind of know what our offspring percentages need to be. So if we do the Punnett squares for these, then we can know what type of ratio we need to look for to see what would match. So let's just look at D. D says one parent is heterozygous for flower color. So that means they would be, what letter do I want to use? I'll use a P. I'll try to make my P's different so you can tell the difference. So they were heterozygous. And then the other one was homozygous for the purple, which would be capital P. And if we make our little square, then we go across. That means we would get a big P, big P. We would get a big P and a little p. We would get a big P and a big P. And we would get a big P and a little p. So this would have a dominant in every single offspring, which means that that would overpower that recessive white. So this would be 100% would have been purple. So D cannot be correct here because it does not match the split of our offspring, which needs to be 75 and 25. So we know to get that recessive, we have to have two recessives coming together. So there has to be both parents. I apologize for the background noise. My son has been homesick, and so please excuse that. So in order to get that white, we have to have two recessives come together. The only way for the two recessives to come together is if both parents have a recessive. So that means that A is really the only possible choice here. Both parents have to be hiding a recessive. They both have to be heterozygous so that we can... Excuse me for a minute. Okay, I'm sorry. I apologize for that. All right, so A, again, like I said, is the only correct answer because that is the only way that we can get a recessive to be hidden in both of the parents so that it can come together in the offspring. So here's that Punnett square that I've shown here. Again, like I said, the easiest thing to start with those types of questions where you're kind of working backwards, trying to find the parents from what they give you as the offspring, is to first find out what percentage of each of the offspring phenotype is. So the best way to do that is to add up your total offspring and then find the percent that each phenotype represents. So in this case, 89 of the were purple, so that was about 75%. It was a little less, 74. And then we could do that with the 31, and that would end up being close to 26 probably. So that is very close to that 75-25 ratio. Because remember, in nature, it's never going to be exact. We're always going to have some difference, but as long as it's close to those main ratios, we can use it to guide us. And then when we do the Punnett square, we could see when both of them are heterozygous. In this case, both parents are heterozygous with the lowercase w hidden. We can see that we are able to get a 25% chance of that white and then the rest being purple. So that's why the answer should have been A. Does that make sense? Okay, great. All right. And I had a blank page in here just because, like, don't be afraid. If you have to go through each of those answers, like it gives you 4 different possible combinations of parents here. If you have to draw out the Punnett squares for each of those to see which one matches, take the time to do that. On this EOC for the biology, you have plenty of time. I would say way more time than is really needed to get through the exam for most students. I've proctored the test quite a few times, and I've never had a student run out of time. So that just kind of shows you, and that, you know, on all levels. So take the time if you need to, to work out the problem and work each of them out until you find one that matches what the question's giving you. Don't be afraid to do that, and because you've got the time to do it. Okay, you're not going to run out of time if you were to sit down and do that, because not every question is going to require you to do something like that. All right, here we're going to DNA. Let's do a question about DNA. This one says a codon is a sequence of 3 nucleotides that code for an amino acid. If the DNA sequence shown here goes through transcription and translation, how many amino acids will result from this sequence? So here's the DNA sequence up here. It's kind of hidden. To me, I would think they should put it at the end, but anyways, they put it at the beginning. So based on this, how many amino acids will we get from that particular strand? If we know that a codon is a sequence of 3, and 1 codon codes for 1 amino acid. D, excellent, perfect. We would get 3. Okay, and that's because if we break this up into 3 blocks, every 3 amino acids is every 3 nucleotides is 1 amino acid. So we have 1, 2, and then 3. And this is just an overall view of that process from DNA to the amino acids. So remember, the DNA forms that template strand. The messenger RNA is going to complement it, substituting where an A would normally match with a T for thymine and DNA. We substitute that out with a U for uracil on the RNA. And then every 3 represents 1 amino acid, and we link those together to form our protein. All right, another DNA question here. It says a cell is replicating its DNA. Part of the DNA strand reads ACC, GTAC. The new strand built off this section, when it's replicating, so this is just making a copy of it, the new strand reads TGG, and so this would complement, okay, and that's correct. T complements A, G complements C, G complements C again. Then we have an AAT. This is where there's a mistake. The first A there is matching up with the G, and it should have been a C as a complement. And then the other ones are correct, and then the G matches with the C. So it all is complemented and basically replicated correctly, except for that one A should have actually been a C. So what effect might this have when the DNA replicates in the future? So when this new DNA goes to replicate in the future, which of these is going to be correct? Is the cell having the new DNA going to look and operate differently? Is the only section of DNA without mistakes would be used in the future, so the DNA strand will shorten? Will the DNA be exactly like the original, since the original strand of DNA is used as a template? Or D, will the strand used as a template to build the new one differ? I think we got one option here. Anyone have any other guesses on this one? Okay, so we have kind of going between A and D is what it looks like you guys are deciding between. We know that you're not going to just only use the sections without mistakes. That doesn't happen. The DNA will possibly shorten over time. That's actually just part of the aging process, but it's not because certain sections aren't used because they don't have mistakes. So that one's a definite no. C is a definite no because we don't just use the original strand as the template each time. Okay, every time a DNA is replicated, it's going to have part of the old and part new, and then that means that that new part will get used again to replicate in the future. So between A or D. For A, cells having the new DNA version will look or operate differently from cells with the original DNA. That is possible. The reason this is not correct is because the question is asking what effect might this have when the DNA replicates in the future. It's not asking you what effect this will have on the cell. It's asking you what effect it will have when it starts to replicate again. And this answer is not talking about replication, it's talking about the cell and how it will act. So that is why A is not correct. So this was a little bit of a tricky one because it does give you an answer there that is possibly a correct answer, just not for the question being asked. So that's why you have to be very careful when reading these questions and make sure you are referring your answer to what the question's asking. And in this case, it's asking about the replication process in the future, not how the cell is going to operate. So D is our correct answer here. When that new strand that has that mistake is used as a template to build again, to build another strand for another replication, it will differ from the original, and that will then pass that mistake on to the next cell and the next cell and the next cell. So let's just take a look here at some different types of mutations that can occur because of those types of things. So this top one here is our normal transcription where the DNA is no mistakes. So the messenger RNA that forms is where it's supposed to be, and those are the amino acids that were supposed to be translated from that DNA. In a substitution, which is what we saw happening in the question we just did, we have one of the bases being switched out. So in this case, it is actually here. This is a T instead of the C that it was in the original. So that now makes this mRNA nucleotide a different nucleotide, which is what was happening in that question before, and therefore we have a whole new amino acid being placed in this protein, which is going to make the protein not work the way it's supposed to. So that's called a substitution. An insertion is when the DNA, when it's replicating itself, puts in an extra nucleotide than what should have been there. So in this case, an extra C was copied in instead of just the one C. An extra C was put in, so we actually have a whole extra nucleotide. So that puts in an extra G on our messenger RNA. So that's called a frame shift, or in this case, an insertion. Because of a frame shift, it's going to cause all of our amino acid codons to shift over. So now, what would have been the GAA, there's an extra G in here, so now it's GGA, giving us a glycine, and now it's switching the amino acid after that as well, because that's been shifted over. So every amino acid from this point forward is possibly going to be a different one, and that is going to really mess up that protein. A deletion is the same way. A deletion is where when the DNA was making copies of itself, it missed one of the nucleotides. So in this case, we missed one of the A's on the DNA. That A was taken out. It should be up here. It should have been in there. So the A was taken out. So instead of a uracil being placed in here, it's no longer there. And so it shifts again. It's shifting everything over, therefore giving us all different amino acids that should have been there and messing up that protein. Okay? So that's some examples of how when DNA replicates itself, we can get mutations that will cause defects in the proteins eventually. All right. Any questions about any of the DNA or genetics that we just covered, or anything Mrs. Tyson covered earlier this week? No? All right. Well, thank you guys. I'm sorry about those technical difficulties at the beginning and having you move over here, but I'm glad we were able to still get this done. If you, I appreciate you guys joining us and joining me today. We will be doing this again next week, and hopefully it will still be working. We'll be able to get Miss Tyson's classroom working fine. So plan on it being in her classroom next week. If for some reason it's not working, then I will, direct you back over to mine. But thank you again for joining me. You guys have a great day, okay? All right, bye-bye.